Okay, hello everyone, and welcome to this workshop, which is part of the Early Researchers Seminar organized by the Cambridge Reproduction SRI. The Cambridge Reproduction SRI is an interdisciplinary initiative that brings together researchers from across the university who have an interest in any aspect of reproduction, everything from science, technology, and medicine to arts and humanities. If you are not yet a member, you can download the membership form from the website. Now, I would like to start by thanking the volunteer coordinators for organizing the series, and indeed to all speakers for volunteering their time to be here with us. The seminars are held online on the second Thursday of each month at 1 p.m. And if anyone here who is a postgraduate student or a postdoc would like to take part, please get in touch with Christina. So my name is Rumiana Yotova, and I am from the Faculty of Law, where I work on international biomedical law and human rights. And it is my great pleasure to welcome our two speakers today. Our first presentation is going to be given by Suhan, who has an undergraduate degree, MSc and PhD from New Zealand. She came in Cambridge in September 2019 as a postdoc to work with Professor Herbison, and her lab and herself have a long-standing interest in the brain control of fertility. So Sue is going to talk about the shedding light on the GnRH pulse generator.
Thank you very much for the introduction. And thank you very much for the opportunity to talk at this Reproduction SRI Early Career Researcher Seminar Series. I am a neuroendocrinologist studying brain control of hormones as a postdoc in Herbison Laboratory as introduced at PDN department. And as being introduced, I'm particularly interested in how brain controls fertility. Fertility affects our lives and community. In fact, infertility is the major issue worldwide. In UK, 1 in every 7 couples are diagnosed as infertile, and many women indicated that coping with infertility is one of the most upsetting experiences in their lives. It is usually viewed that infertility is the problem of women, but it's not really the case. Indeed, infertility affects the women and men equally. Therefore, it is really important to understand the normal biology of normal fertility in both sexes and what can go wrong that could contribute to infertility or subfertility. So when you think about fertility in a biological term, this is probably the #1 image that would pop up in your head. Well-known reproductive organs, testes in males and ovaries in females. And although it is true that these are the first reproductive organs in fertility, they are not the only ones. And I think you can probably guess the organ that I'm going to introduce. It is brain. Brain is really important for normal fertility. And why should we put brain in charge? Because brain is able to perceive those outside cues such as environment, for example, nutrient factors, physiological status, and psychological status, and is able to integrate that information, especially within the area of hypothalamus in the brain. And it can provide the appropriate and pivotal commandment to the testes and ovaries, the reproductive organs, in the means of gonadotropin hormones. So let's zoom into this hypothalamus area where the master output brain cells regulating fertility is located. They are gonadotropin-releasing hormone, or GnRH neurons, shown in green here. And they are scattered in the hypothalamus, and they do send their long projections down to this median eminence, where they release GnRH into the portovasculature. In the pituitary, there are gonadotroph cells that can respond to GnRH, and they stimulate the secretion of luteinizing hormone, LH, follicle-stimulating hormone, FSH, into the bloodstream. So they can then act onto the gonads, testes, and ovaries to produce the sex steroid hormones such as estrogen, progesterone in females, and testosterone in males. And these sex steroids are really important in spermatogenesis in males and ovulation in females, but they are also very important in feedback onto the GnRH neuron system back to the brain to further regulate the secretory pattern of these hormones. So this very tightly controlled loop is called hypothalamo-pituitary gonadal, or HPG axis, which is the critical axis controlling the normal reproduction. So one of the very interesting and important features of GnRH neurons is that they produce and secrete GnRH in a pulsatile fashion. And once released, that pattern of release is faithfully mirrored by the pattern of LH and FSH. And because GnRH is very hard to be measured in blood, and also it has a very short half-life in the blood, we usually use LH as an indication of GnRH pulse or GnRH secretory pattern. So let's have a look at the GnRH pulse pattern in adult reproduction. You can see here in males, the pulsatile or episodic secretion of GnRH is predominant, whereas the females have these pulsatile secretions in follicular and luteal phase. But in the middle of the cycle, there's a massive outpouring of GnRH that leads to LH surge that causes ovulation. But except for this phase, the predominant phase or mode of GnRH action is the pulsatile secretion. This pulsatile profile of GnRH and LH secretion seems to be very important in maintaining normal reproductive function, as some reproductive disorders exhibit differential patterns of LH secretion. For example, here I show you the LH profiles from polycystic ovarian syndrome patients or PCOS patients. And compared to the normal healthy women with nice slow pulses, these women do exhibit high levels of LH established by high frequency of the LH pulses. On the other side of the spectrum sits women suffering from hypothalamic amenorrhea, where their pulses are a lot slower when compared to the, again, normal healthy subjects. So when we have a look at the GnRH neurons in brain, here I'm showing you the sagittal section through mouse brain, and the GnRH neurons are stained in black. You can see that these neurons are not really confined within one area or one nucleus, but are kind of scattered throughout the hypothalamus. So for the GnRH neurons to be secreting GnRH in a pulsatile fashion, these neurons must somehow to be synchronized to generate pulses of LH, GnRH and LH. And the question is how this is achieved. So in large, there are two possible scenarios. One is that GnRH neurons have the intrinsic property for them to be able to form a community themselves to be synchronized and produce the GnRH and secrete them in a pulsatile fashion. But more and more studies have suggested for the existence of the extrinsic pulse generator. And for the interest in time, I can't really explain to you all the studies that's shown there, but long story short is that these extrinsic pulse generators may be able to control GnRH neurons either at the cell body or either at the projection level down to the median eminence to control their activity. So if such extrinsic pulse generator exists, what's the identity of them? Among many neurotransmitters and proteins, peptides that can regulate or manipulate GnRH neurons, the most potent or powerful controller of GnRH neurons is called kisspeptin. So this protein kisspeptin was first discovered by a group working in Hershey, Pennsylvania in 1996. So that's why it's named after the city's chocolate kisses, which are made in Hershey. So the discovery of this kisspeptin has really revolutionized our understanding of fertility. Again, with interest in time, I can't explain everything, but long story short is that kisspeptin is really essential in normal reproduction, as loss of its receptor results in hypogonadotropic hypogonadism, and subjects fail to reach puberty and become infertile. So the number of studies afterwards have really emphasized its critical role in reproductive function. And indeed, kisspeptin action at the GnRH neuron itself seems to be essential for pulsatile LH secretion. So here I'm showing you two examples of a pulse profile from normal male showing a nice pulsatile fashion, and the male with kisspeptin receptor mutation. So the kisspeptin is not able to act onto its receptor anymore, and you can see these pulses of LH is completely gone, and the LH level is very low. The same phenomenon happens in mice. So when people engineer the mice that has no kisspeptin receptor, they found that in females have very small ovaries, males have very small testes compared to the normal mice. And not only that, when they measure the LH profiles, the normal ones have nice LH pulses. Compared to that, the kisspeptin receptor null mice has no pulses of LH whatsoever. They were able to rescue back the kisspeptin receptors only to the GnRH neurons and found that the ovaries and testes size has come back to normal and the fecundity has returned to the normal state. And also, they found that the LH pulses have become normal as well. So this really highlights the crucial action of kisspeptin on GnRH neurons for the pulsatile LH secretion. So there are more than one area where kisspeptin neurons are found. There are two major populations of kisspeptin neurons. Here I'm showing you a sagittal section through mouse brain again, and there are two populations highlighted in blue and red. So the pre-optic kisspeptin neurons are critically involved in surge generation, and many studies have shown that, so it's also known as a surge generator. Compared to that, not many studies have really focused on to the arcuate kisspeptin neurons, which is located at the more back of the brain, but studies suggested they might be critically involved in pulse generator, but there had been no direct evidence showing it up until a few years ago. So that really came down to our simple and straightforward question: are these arcuate kisspeptin neurons the GnRH pulse generator? So to study that, we really need to be able to see what these arcuate neurons are doing in an animal. So we need to be really monitor these neurons specifically. And also, we also need to be able to manipulate the activity of these neurons, either stimulating them or inhibiting them, and we can relate them to the pattern of LH as an output. So to study that, I used all-optical approach to monitor and manipulate kisspeptin neurons using light and light-sensitive proteins. So the first one I'm going to explain is the monitoring of arcuate kisspeptin neurons using the technique called fibre photometry. Fibre photometry uses a biosensor called GCAMP. It is essentially modified green fluorescent protein, GFP, and as its name indicates, it's the protein that fluoresces in green. And this GCAMP does not normally fluoresce, but in the presence of calcium, it can bind to a subunit of this GFP, GCAMP, and then it becomes fluorescent. So because cell activation is closely associated with intracellular calcium rise, we can use GCAMP as a proxy of neural activation. So we used a genetically engineered mouse line, so we can express this GCAMP into the arcuate nucleus specifically to kisspeptin neurons using viral vector. So you can see this viral vector has the GCAMP in it. So by injection, we can transfect kisspeptin neurons with the GCAMP. So here is our kisspeptin neurons transfected with GCAMP, and we can now bring down a very thin fiber down to the arcuate nucleus to provide the blue light excitation. And this fiber is then connected to the recording system, so we can provide light, and at the same time we can record or measure this fluorescent level coming from this GChem down to, up to the same fiber. So for example, the fluorescent level will be very low when the cell is not active. And when the cell becomes active, we can see it here as the fluorescent level rises. And we can use that as a proxy of neural activation. So here I'm just highlighting, showing the coronal section through mouse brain, showing where the fiber optic was placed at the very base of the brain where the kisspeptin neurons are. And I can record the fluorescent levels or activity in a freely behaving mouse. I'm showing you an example of an activity of arcuate kisspeptin neurons in gonadectomized male mouse first. Again, here on the y-axis, I have the fluorescent level over time in minutes. You can see that these neurons indeed exhibit episodes of synchronized activity. When we have a look at this activity closely, each episode shows a rise in the signal over 20 seconds, followed by a slow recovery of 50 seconds. So the whole episode lasts just over a minute. And these episodes occurred as frequently as 9 minutes in gonadectomized mice. So I was very happy to see this when I first was able to record this. This was the first direct evidence showing that kisspeptin neurons in the arcuate nucleus provide synchronized activity. However, the more important question is whether this kisspeptin activity is driving GnRH, thus LH secretion. So what I did was to combine this photometry recording with tail tip bleeding blood sampling method. So the thing is, I cut a very tiny bit of tail of a mouse, and I can collect the blood, very small amount of blood, and pipette it out, and we can measure the LH concentration in the blood. That can be done during the photometry recording. So here I'm showing you two examples of the photometry recording again, showing a very nice synchronized activity of kisspeptin neurons in two different mice. When I measured the LH concentration, I could see a nice pulsatile secretion of LH, but more importantly, they matched exactly same as the kisspeptin neuron activity. Indeed, these kisspeptin neuron activity preceded this LH pulse about 4 to 5 minutes before this LH peak occurred. So this result really shows that arcuate kisspeptin neuron activity drives the pulsatile LH secretion. So this was really nice to see what endogenous kisspeptin neurons are doing, and it has a high correlation or perfect correlation with the LH pulses. But now, is this kisspeptin neuron activity sufficient to drive the LH pulses? So to do that, I decided to manipulate these neuron activity using optogenetics. Optogenetics use light-sensitive channels or pumps. So first of all, I used the stimulatory optogenetics by using a light-sensitive channel called channel rhodopsin. So just in short, this channel is a cation channel, and it only responds to blue light. And when activated, it allows for the cation ions to flow into the cell, therefore activate the cell. So I employ the same method as before, but this time use a viral vector that carries channel rhodopsin into the arcuate nucleus. So here our kisspeptin neurons are expressing channel rhodopsin, and this time with the fiber, I delivered pulses of blue light lasting for 1 minute, repeated for 30 minutes. I chose 1 minute because with the endogenous kisspeptin activity, it just lasted over a minute. So by mimicking what the endogenous kisspeptin activity is doing, I try to find out whether stimulating this kisspeptin activity can drive the LH pulses. So here is just, again, the summary of the experimental setup. Fiber optic is down to the arcuate nucleus with the kisspeptin neurons expressing channel rhodopsin. And this time I'm measuring the blood, I'm collecting the blood for the LH measurement during the stimulation period. Here I'm showing you the LH profile or concentration on the y-axis over time in minutes. And these blue bars indicate where the stimulation of channel rhodopsin or kisspeptin neurons were given for 1 minute each. And you can see that every time we turn on the kisspeptin neurons, there is a pulse of LH evoked. And indeed, this pulses of LH seems to be very similar to that of an endogenous pulse when overlaid indicated in red here. Because of the blood sampling rate was different in these two experiments, I couldn't really do a statistical comparison, but you can probably appreciate the shape of the pulses in both cases are pretty similar. So it was really great to see that we can selectively activate kisspeptin neurons in the arcuate, which is sufficient to generate LH pulses. But this does not necessarily mean the necessity of these neurons for LH pulse generation. So we decided to utilize another optogenetic technique, but this time using an inhibitory tool to silence or inhibit the activity of arcuate kisspeptin neurons and see whether it can suppress endogenous LH pulses in freely behaving mice. So there are two majorly used inhibitory opsins or pumps called halorhodopsin and arcuate. We did experiments using both, but we decided to, I decided to just show you one using the arcuate for the interest in time. So arcuate responds only to the yellow light. And when turned on, it actually provides the cation ion going out of the cell, therefore inhibiting the cell. So I used the same method again, but this time using the virus that carries arcuate. And this time we provided the green light for half an hour and see what happens to the endogenous LH pulses. And this particular experiment was led by Dr. Jenny Clarkson, who was my colleague in the Herbison laboratory in New Zealand. So the results show that this endogenous pulses of LH shown in these two examples were greatly diminished during this period of inhibition of kisspeptin neurons. So for summary, the pulse amplitude and the number of pulses were both greatly diminished by inhibiting the kisspeptin neurons, which suggests or strongly indicates that kisspeptin neuron is necessary for pulse generation. So just to summarize and conclude, I've shown you that kisspeptin neurons in the arcuate nucleus show synchronized episodic activity. It correlates perfectly with LH pulses. And by providing 1 minute activation, we can evoke LH pulses. More importantly, we can inhibit these cells that suppresses LH pulses, which means that these neurons are absolutely necessary for normal pulses to occur. So the take-home message would be the arcuate kisspeptin neurons are the GnRH pulse generator in mice. So I'm just gonna show you my last slide, the current work and the future direction. We're doing more photometry recordings in male and female mice and also a model of PCOS just to see what the pulse generator activity does during the disease state. We also use more technique called microendoscope to see what this individual kisspeptin neuron is doing. And we're also doing some investigation under the mechanism underlying this kisspeptin population synchronization. So with that, I would like to finish by thanking the Herbison Lab, both in New Zealand and Cambridge, and the collaborators, and all the funding bodies back in New Zealand and here in UK as well. And thank you for listening. I'll be happy to take questions.